Hello, welcome to the Kansas State Veterinary Diagnostic Laboratory YouTube channel. All samples for diagnostic testing should be packed using 3 layers of packaging material. This video discusses more specific recommendations for particular sample types. When submitting serum samples, please allow the blood to clot, spin down the sample, and collect the serum for submission. This will allow you to assess the degree of hemolysis and/or lipemia present and results in a stable sample. Multiple blood tubes are packed to prevent them from banging together in transit. Individually wrapping the tubes, using a styrofoam tube mailer, or reusing the packaging that the tubes came in will accomplish this. Do not send any blood samples in a syringe with or without a needle attached. Ice packs should be included in warm weather. Please do not send ice cubes. A styrofoam cooler or other insulating materials will help maintain the proper temperature. Blood and serum samples are considered potentially infectious, and the outer packaging should be labeled with UN-3373, an animal diagnostic or clinical specimen. If submitting tissues in formalin, please use a sturdy plastic wide-mouth jar with a screw top as the primary container. Tupperware, Ziploc bags, and Whirlpack bags are not designed for, nor are they capable of containing liquids through the rigors of transportation. The jar must be labeled indicating that it contains formalin. Wrap the jar in absorbent material and place it in a well-sealing plastic bag. There is no need to send fixed tissues on ice packs. In fact, formalin penetrates better at room temperature. Fixed specimens should be protected from freezing. Therefore, in winter, a cooler should be used to protect the sample from extreme cold. Fixed tissues are not considered potentially infectious and do not require UN-3373 labeling. The outer packaging should be labeled clinical specimen. These samples should be collected as cleanly as possible into clean or sterile containers. Exam gloves and OB sleeves are not appropriate containers. The primary container or swab should be placed in a baggie or Whirlpack. These samples should be protected from temperature extremes with a styrofoam cooler. Ice packs should be provided to achieve a refrigerator-like environment. Ideally, an equal volume of ice packs and tissue is used. If sending a large volume of tissue, it is best to chill the tissue overnight in the refrigerator prior to shipping it with ice packs for overnight delivery. The box should be labeled with UN-3373, an animal diagnostic or clinical specimen, as these samples are potentially infectious. These samples are fragile. They should be placed in a holder designed to prevent breakage. Cardboard, plastic, and styrofoam slide holders are all acceptable. If your slide holder has a snap top, please tape it closed to prevent it from popping open in transit. Slides should not be sent on ice packs, as the condensation may rinse the cells off the slides. Cytology slides should also be protected from formalin and its fumes by individually bagging the slides and any fixed tissues sent with them. Dried slides are not considered potentially infectious and do not require UN-3373 labeling. The outer packaging should be labeled animal medical or clinical specimens. Thank you for tuning in to the Kansas State Veterinary Diagnostic Laboratory YouTube channel. If you have questions about this video or any of our services, please visit us at www.ksbdl.org.